My name is Roland Gamsjaeger. I'm from Austria originally. I'm born in Austria. I did my degree in Austria. So I actually studied chemistry. I did a technical chemistry degree first, and then kind of at the end of the degree I started to have develop an interest in biology, and we had this unit called biophysics that I then studied at the end, and then I ended up doing my PhD in there, and that really sparked my interest into biology in general and mechanisms, molecular mechanisms in cell, how cells work. And that's how it started. And then I did a PhD, as I said, and then after the PhD I did a postdoc, which is what you do after your PhD. So you normally go overseas. That's what people mostly do. And so I decided to come to Australia because there's good science here and the climate is great, and for other reasons. And specifically to the place at the University of Sydney, because I was doing a lot of online searching. I wanted to go to the right place with the best science and combined, as I said, with other lifestyle factors, and that led me to go to this lab at the University of Sydney. And then I've done a postdoc there for 7-8 years before moving to Western Sydney. I think it started at school, really. I started kind of at the end, before what in Austria is called Matura, or it's the HSC. That's where I kind of developed an interest in science in general, chemistry in particular. So then the question was, would I study chemistry or informatics? That was always the choice between the two, but then I decided for chemistry. That's why I then did my chemistry degree. And during that degree, obviously still loving science as it is, and I then kind of more moved into the biology. So I was still interested in chemistry, but more into the chemistry, what happens actually in the cell, in the biology, what's driving the life really, what's the basis of the molecular basis of life, those type of questions. That's when it started anyway. There would be more than one actually. I had a really good teacher in school that was very passionate about some areas of science, and then later on during my PhD I had a really good mentor as well that kind of helped me along. And then probably the most important scientist I would say was when I did my postdoc at Sydney Uni. That was really a person that influenced me a lot, and that made me think, okay, I want to do not just science, but I want to stay in academia. I think I would mostly say it's driven by the literature really. Like I work in a field, specifically DNA repair and chromatin remodeling, so some really basic mechanisms that have to work in the cell, otherwise the cell is not working, basically. And within that area, I'm very much driven by questions that we want to answer, and what's out there in the literature, what's already known, and where can we contribute to things that are not known, and that's where it really drives. So we have a set of methodologies that we use. So we don't have unlimited resources. We have only a certain limited set of resources and methods that we can use. But we do work with other people, and that's part of the science. But specifically, the driving force is probably what's known in the literature, and that comes from the publications, from the science. I seek advice from my collaborators, from people that I work with, but specifically the decisions are probably being made by myself and the lab that I work with, my students, and I work with another academics very closely at Western Sydney as well. So that's really the driving force there. So we come together, we talk specifically with my colleagues here, but really the decisions are then made by us, more or less, where we're going and what we can achieve. I think it's the question of what's happening in the cells. That's really what drives me all the time, like what's really going on. And I don't know, maybe it's my character, but I like to dig into things. I like to understand things, the small things, the molecular things. That's where my approach is, probably coming from my chemistry background as well. So I like to dig in, I like to see what's happening between the molecules, what's interacting with what, and how does it do that, and that's really where it comes from. We look at what's there, and then we look at how we can solve some of the questions that we're asking. And most importantly, as I said, we would look in the literature and see if there's already people working on that, because there's quite a bit of competition in the science field, and that's what's driving it partly, but also what's then if there's already someone working on it, I would say, okay, we're not working on it because We don't want to compete with some big labs. For example, in the US, they have 5, 10 postdocs working and lots of money. We don't have that here. Right? We have some limited resources. We have some resources. We have good resources. We have good students. But we really have to be careful what it is that we're working on, so that we can make a contribution. Because what can happen, of course, is that you work on something and someone else works on something, and if they publish that first, then you basically have, even though you've done the same work, it's kind of not the same acknowledgement because it's already published. That makes sense. So it would depend very much on what it is specifically that then we have to look what's known already, who is working on it, what's out there in the literature, where can we make our contributions with our set of methods that we have. We go to conferences, I exchange ideas with people, I listen to talks. That's a way of communicating. I have collaborators, I have a network of friends, of people that I know that can tell me whether other people work on certain things or not. It's very much like that. So conferences, the literature that's published. First of all, we would ask the question. So we would have a hypothesis. We would say, okay, this might work like this. To prove that, as I say, we probably ask the question so that we could solve it with the methods that we have. If we don't have the methods to solve it, we could ask the question, but we have to ask someone else to help us with. If it's specifically about what we can do, we can say, okay, we've got this set of methods that we can use, and that's what we do next. So we then design experiments that make sense to then answer that question. So we would set up a whole set of experiments which we think we have to do, and then the next thing would be who is going to do it. So we have to find someone that does it. In my case, it's the PhD students, or we have a postdoc as well. So it's also managing who is doing it. So that would include then determining, okay, this person is more suitable to do that, this person is more suitable to that. But in our lab, we basically take the approach that everyone knows sort of almost everything. And people have still specific skills, but the basis of everything, the basis of all the methods everyone is aware of. So it would then say, okay, do you have time to do this? Ask the student, or are you interested in doing this, and then design a project around that, like a small PhD would be part of a PhD project, or part of we don't have honors anymore, but master's project. So that would be the next step after thinking which experiments we actually need to do. The most challenging part is getting funding, to be honest. That's really the most challenging funding. In the current environment in Australia, the funding is not easy to get to, especially government funding. There's a lot of competition, and we have big groups in Australia that have, as I said, they're well funded, they have millions of dollars research money, they have lots of postdocs, lots of students. We are a smaller group, so we have to really be careful with that. We have to make sure that's why we said we have limited resources that we have to use efficiently. So we have to plan our experiments wisely. We have to use the synergies that we have in the lab, because the science that we do in the lab is very, we need consumables. We need basic consumables. So it's not, you can go out and do a survey or whatever, it's not that easy. We really need to buy consumables because we have to do experiments. So that's cost money, and the funding is very constrained in Australia at the moment. Most of the data we're collecting is quantitative, so we're doing experiments, we're getting results, and then we're analyzing those. So we're using statistics to do that. And in biology especially, it's important to have a decent number of experiments. So when I say number of experiments, you repeat an experiment 3 or 5 times to get to a conclusion. And it's often a lot of effort, but you have to do it. If you do an experiment once, you can get one result. If you do another time, you get another. So it can vary a lot because there's a lot of parameters. It's not a simple thing to do. So you have to do it many, many times. That's really the basis. My specialty is looking at the structure of protein molecules. So starting what the three-dimensional structure is of protein molecules. So basically, what shape they take on in the cell to perform the different functions. And I use a method called NMR to do that, nucleic magnetic resonance spectroscopy. So that involves using a big machine, a big magnet, a bit like when you pushed into the MRI magnet, similar principle. And that's requiring a lot of protein, so we have to make the protein first and then analyze the protein. And so we get a lot of data from that. Then we run experiments that can take 2 weeks, and then basically you spend 3 months analyzing those data, going through this data, using software packages, using statistics, to then at the end arrive at the structure of the protein. It's a very elaborate process and needs to be checked and rechecked. You go back and forth, so it's very much an iterative process. We're using a lot of biophysical methods. So that means we combine physical methods with biology. So we're using, a system is the biology that we study, but we're using spectroscopy, for example, to study, we look at protein concentrations by spectroscopy, we look at folding by NMR, CD, we use CD, we use also biochemical methods, so we run gels, run protein gels to verify if the protein is clean or not. So it's very much a combination between biochemical and biophysical methods. And it's like I said, it's a suite of methods that we use and that are complementary. So one thing will tell us one thing, the other method will tell us a similar thing. If that doesn't happen, then we know something is wrong. And we use maybe a third method to actually verify that's what it is. Because an example is often proteins are, the publications out there that claim that one protein interacts with another protein, and we want to actually verify that. It often turns out that's not happening because these experiments were done in cells, and it's not always in cells, and people find things in cells that don't work when you then actually look at those two things in isolation. So we verify that with different methods whether there's actually an interaction, and if we don't see an interaction, clearly that's not happening then. I mean, one of the things we use a lot is fitting. So we have an example is when we calculate the protein affinity. We would use different concentrations of the protein, different concentration of the binding partner. There could be DNA or another protein. Then we would look at that, we would plot that in programs, in graphical programs we plot, and then we would fit those data. So fitting is something that we use a lot. And obviously if you do fitting, you need a lot of, the number of experiments needs to be large enough to be confident that the fitting that you see is actually real and not some artificial fit, because sometimes you can fit everything with everything, right? You have to really, has to be a basis to it. There's a huge amount of data out there already on the internet, in databases, that where people solve protein structures and they've been looked at and verified. So it's very much the way of doing things these days. So you look at that in isolation first, then you solve the structure and you publish that structure, and then you look at whether the structure, what the function of the structure is. But in most cases, when you then verify it, say 99% of the cases, the structure is the structure defined in isolation is the same as when it's in cells or in a whole organism. But you do need to do these experiments, and you do them in cells. I would say this is a large data set that consists of many different parts, but overall then is going to be used to, like I said, for example, in my case, then solve the protein structure, so determine what this protein molecule looks like in 3D space. The long-term aim is to understand how certain diseases work, and in our case it's mostly cancer. So it's really in cancer lots of things go wrong, and if you understand the molecular basis of those systems that we're studying, for example, you can then develop drugs, for example, that can help you fight those diseases, and that's what we are now working towards. So I mentioned funding is a big issue now, government funding in Australia. So we're now trying to shift our focus a little bit on industry as well. So we're trying to work with biotech companies that are around, that we can help with, that we can support things that they're interested in, and we can help them. And at the same time, we're also trying to develop our own research and trying to develop some of those molecular mechanisms. We're trying to actually translate them into developing a drug, for example. Because another example is if you know how two things interact, you can develop a drug that blocks that interaction. So you can then specifically block that. And if that's important in a disease, you can obviously use that. And that's already been used. So we're not inventing this. There are already drugs in our particular system that we're studying, which is DNA repair, that people have actually gone all the way through the trials. And then there are actually now cancer drugs available which are based on the principle. That's where the publication comes in, going to conferences, talking to people, but it's mostly through publishing. So publishing is very important. You need to, once you're confident you have a set of data that confirms or answers one of the questions that you're answering, you would then write something up and then try to publish that and try to submit that to the different types of journals out there that would accept it, and it's another story for another day. I'm talking about years in my area, in biology. I'm talking about years. So once you have an idea, you design, we talked before, design the experiments, then you find the right people to do them. It would take a year to do them, and then maybe take another half a year to get published, because it's also a challenge, as I mentioned. So you'd say 1 to 2 years, and in some cases maybe 5 or 6 years, in some cases maybe only 1 year. But that's what the time frame. So I think in biology it takes a little bit longer. If you're talking about areas like chemistry, maybe it's faster, but in our area it takes longer because those experiments have to be verified and have to be done, and often things don't work as you want them to work. It's not a straightforward thing, and you do a lot of troubleshooting, really. That's what our students do a lot. The main advice that I would give them is don't look where your friends go to uni, look at what you're interested in, and then look at the academics themselves that are at the universities doing the research. Look at their papers. I know this might be hard for year 12 students, but there is a database out that you can look at. You can look at those papers, or you can at least have an idea what is published, what is researched, and then go specifically to that lab, to that area where you're interested in. And don't choose the uni based on their name or on where their friends go. As I said, choose it because they're doing some interesting stuff that you're interested in, and go there and study it with the aim of then working with those people at the end.